Good afternoon. I want to thank the organizers for inviting me to talk in this meeting. After so many wonderful talks, I wonder if there's still anything more to add. But anyhow, it's also very exciting. This is also my first talk after about a year and a half at home. And I'm almost tempted to thank you for opening your cameras, because I can see everybody here. So in the lab, we work on clinical questions. We try to help the clinical team to reach the right decisions. And we combine it with bioinformatics or analysis of the data. And we also have biology in the lab where we can dig into the questions and see whether maybe we can understand the biological process as well. So if we have a patient or an individual, we work on the DNA, which is probably the past, the present, and also the future of your medical records. RNA, which is much more volatile. So if I am under stress now, the RNA is elevated in my body and we can measure it. The DNA is not changed right now. Medical records, which we could look into, tap into, and understand. And under other, we heard many lectures today. It could be microbiome, it could be the immune system, it could be lifestyles, it could be many other measurements that we can take. But I want to start with a very basic question. Why digital medicine? And maybe it's a good way for a closure here if we start back hundreds of years ago. So if we look at the life expectancy increase in the past 200 years, from an average of about 35 to more than 80. If we look at the population growth in the past 200 years only, it increased from less than 1 billion to about 8 billion. So how will we be able to treat so many people and not only extend their life, but also their quality of life? And the answer, of course, is assimilation of new technologies. If we look here, we know again that in the past 10 and 20 years, there's a major adaptations of new technology. And if there's something good, it's assimilated or adapted very rapidly. And we're playing on these rules, and we want to use them as well for our advantage. If you work on genomics, you always show this graph in lectures. So this is Moore's law, the doubling of computer power every 18 months. And you can plot on the same graph how much or how often or how easy it is to sequence the human DNA. And you can see it here decreasing exponentially on an exponential graph. And we do it usually with the first generation, with the second generation, the NGS here in the center. And we know we're just about to add on additional machines, not only the Oxford Nanopore machine you can see here, but additional machines that will help us decrease the a cost of sequencing of an entire human genome within the next few years, just like we saw it drop down in 2007. So we saw here that many labs collect data, but that's not the trick. The trick is to gain information from it, to turn it into knowledge, and then to bring it to the clinic. And obviously, we're losing a lot of data along the way. And we also heard here that instead of taking care of the sick, we want to take care of healthy people or prevent the diseases, and we want to be reactive and not only proactive. We heard that in several of the lectures. So this is what we do. We combine the clinics with bioinformatics and biology, all of them in our lab. And I'm going to talk about a few studies. We'll go throughout life. We'll talk about pregnancy, rare diseases, and we can't obviously finish a lecture without talking about infections. The first study is on preeclampsia. You'll see on every slide in red, you'll see the student who led this project. This is Liron Yofe, and in white you'll see our collaborators. Preeclampsia is a devastating disease, ra'elet harayon. Many women around the world suffer in Western countries and in developing countries. This is a high risk for the baby and for the mother, and the causes are mostly unknown. We followed thousands of women during pregnancy where the first blood test was taken at week 10. We put them in the freezer. At the end of the pregnancy, most of the pregnancies came out well. A small percentage suffered from preeclampsia. We went back to the freezers of week 10. We took 40 future preeclamptic women, 40 controls. We took about 1 ml of blood. We extracted RNA from these samples. We profiled on NGS 20 million RNA molecules from these women. And Liron did a machine learning algorithm and ran this and came up with 25 non-coding RNA molecules that can predict preeclampsia at a very early stage We'll see more information about it soon. But we went over this and we wanted to understand whether we can feel or understand a bit more about the biology. So one of the RNAs that have changed here is called link HELP. HELP is a severe case of preeclampsia. It is known that when accumulating mutations or polymorphisms at this site, you have an increased risk for preeclampsia. We identified here that also an elevated level of RNA could increase the risk for preeclampsia. We also found several microRNAs, non-coding RNAs floating in our blood that could regulate genes. The first thing we did is we wanted to see whether it could also be predictive at a later stage of pregnancy. You can see here first trimester versus second trimester. The answer is no. It's predictive only in the first trimester. The microRNA downregulates or shuts the expression of anti-angiogenic genes. That means that if it goes down, anti-angiogenic genes goes up. This might indicate of the mechanisms. We want to understand what might cause the upregulation of the microRNA. We took placenta cells. We put them under hypoxia for 48 hours. Then we looked at the expression of the genes. And here you can see again, placenta under hypoxic conditions shows reduced microRNA levels. Here you can see the performance of our algorithms. You can see permutation in red, and you can see the real data in green here. Now, you can discuss whether this is good or not good. It's better than flipping a coin. And the good thing about it is that we proved here that just by looking at RNAs, you could predict preeclampsia. To this now, you can add medical records, you could add additional data, and this is what we're doing now. Another good thing of this label of potential preeclampsia is that a preventative measure is giving aspirin at a low dose, at a daily low dose to these women. And this will prevent the preeclampsia. This led us to another study. The question we asked is, could it be an infection that causes preeclampsia? So this is a study carried with the Galil Medical Center. We received placenta of preeclamptic women and controls. We compared 15 sites on the placenta. We generated an algorithm that could look at infections based on RNA, not based on DNAs. We looked at viruses, we looked at bacteria. The conclusion is there are some differences at different sites of the placenta of viruses and bacteria, but it doesn't separate the preeclamptic ones from the control ones. So in pregnancy, we don't only look at RNA, we also look at DNA. So during the pregnancy, there's certain tests carried out by the woman to make sure that the development is going on as expected. Of course, there's sonography, there's amniocentesis, Mei Shafir, and there's also NIPT. There is some risk in amniocentesis to the baby, to the pregnancy, and current NIPT, which stands for non-invasive prenatal diagnosis, is basically taking a blood test at week 10, and then reading a lot of the chromosomes or a lot of the DNA floating in the blood. And if you see a tiny bit more of chromosome 21 and the mother is healthy, you conclude that this extra chromosome 21 comes from the baby. However, obviously, this is like looking at planet Earth from outer space and trying to figure out if there's another continent. We wanted to increase the resolution here by many folds. We wanted to see point mutations in these very simple blood tests. So what we did is we took a blood test from the dad, from the mom, and then we had a mixture of mom and baby. So from the mother's blood, we could take the PBMCs, and that's the mother's DNA. We can take the plasma, and in the plasma, the ratio is about 9:1. So about 10% of the DNA in the mother's blood is from the embryo at week 10. Now, this is a salad that you have to tell it apart. And what we did is we used a Bayesian model based on several features you can find within these DNA. So if you pick a DNA and it's slightly smaller, you can predict its probability of coming from the embryo and not from the mother. So what you can see here is a density plot. In red, you can see the sizes of the embryonic DNA. In white, you can see the sizes of the mother DNA. It overlaps somewhat, but you can also tell it apart. So if you add these probabilities of various features, of sizes, for example, of GC content, of location in the DNA, you could really get very good statistics on these DNA fragments and figure out whether they came from the embryo or from the mother. So this is just one example of our experiments. Obviously, we did this on real pregnant women. This is a very late week. This is week 38. You can see the depth that we read the DNA is almost 300x. The fetal fraction is very high. 30% is from the embryo, 70% is from the mother. I can tell you that this week we're running a sample from week 7 with much lower fetal fraction, and here we're just comparing the addition of the fragment length to our ability to accurately predict this DNA, whether it came from the embryo or from the mother. This is one example here. We add different features, we take it early in pregnancy, and we actually were able to identify or to read the entire embryonic DNA from beginning to end with every point mutations and every insertion deletions in a week 10 blood test. We are expanding this now and using AI tools in order to better segregate them and also not only to look at point mutations but also chromosomal rearrangements. Now moving on to rare diseases. And for full disclosure, I'm a co-founder of a company that does these rare genetic diseases analysis. If we look at this phenotype, we can figure out what the genotype is. Okay, we know it's trisomy 21. If we look at this phenotype, well, I assume most of you do not know what the genotype is. It's actually Englander syndrome. It's a point mutation from each one of the parents in this gene over here, but there's additional changes that can cause it. It could be also a deletion. It could also be imprinting. So what I want to say here, that standard of care is usually looking at the karyotype, looking at major changes. Is there an extra chromosome? And on the other end, you can also see point mutations in specific genes. You can see this even in preconception when you test the parents before conception. However, in the middle, there are many changes that sometimes are skipped or overlooked. These could be obviously insertion deletions. There's a way to test them, okay? You can test them on microarrays, on arrays, and other ones could be tandem repeats. There's always also ways to measure the tandem repeats. Now, if we can combine them all into one test, actually diagnosis could be using one procedure, one-stop shop, one step that could see all the changes. Now, this is common practice. This is something done, but an emphasis should be made on it that whole genome sequencing, if it's done properly, can see all the changes. Here I'm just showing you an example. There's a proband here and a mother. Every tiny colorful or gray dot is actually a read from the NGS machine here, and you can compare the coverage in that site. I think even can you understand that there's a break here. There's less reads in the center. If there's less reads in the center here, it actually means that there are about 6 or 7 exons missing from this gene here at the bottom. Just another example here. You can see it summarized here. You could really see the drop in whole genome sequencing. You're reading the DNA from beginning to end. The coverage is much better, and you have a lot of information you can gain here. If I would have started this as a clinical case, I would say that it's a 27-year-old female, a diagnostic odyssey of many panels, many mutations, and only after whole genome sequencing you can identify the deletion in a specific gene, which is not visible with any other technology. Also, if you use NGS, which is in a limited span. And now we're moving on to what we've done in the past year, and that's infection. We heard several studies here. Now, many labs have been trying to explain how come there's huge differences between infections in countries and death rates, some of them associated to walking with masks. Here, this is a study published a few months ago. You could see here, they claim that strict rules and abiding to the rules might be associated with infection rate. So on the bottom right-hand side, you can see countries that abide to the regulation. On the top left-hand side, those that do not. And if you want to guess where Israel is, I'll show you where it is. Here it is over here. So they put us not in an attractive place. So this is one way of explaining it. But we try to explain it based on genetics. So we scanned many genes and we try to explain how come there's variability in various death and disease in countries. It might not only be associated with closures and masks and abiding to the rules. We scanned various genes. We stopped on this one and focused on it. This is alpha-1 antitrypsin, AAT, a protein that protects the lungs and liver from damage. It gained many publications in September 11 in the United States, in the terror attack, where some of the firefighters had severe diseases and others did not. So the separation was based on specific mutations. We took these mutations, these frequencies, which indicate deficiency in AAT. We plotted around 70 countries, tens and thousands of people. We see a correlation here. Obviously, this is a hypothesis, but we are running experiments now with hospitals to see whether specific mutations could really explain the severity of the disease and also the levels of AAT in the blood or in the swabs, whether they can explain the severity of the disease. And our initial results indicated the levels of AAT, the RNA levels of AAT in the swabs, could really explain some of these results. We're working with a team at Shamir Medical Hospital with Dr. Adina Bar-Chaim's group, and this is ongoing studies. This is a plot of around 30,000 serology tests. You can see recovered on the left-hand side and vaccinated on the right-hand side. You can see a very interesting distribution here. We are looking at response to infection, not antibody response to vaccinations, but to infections here. You can see separations between severe symptoms and mild symptoms or without symptoms, and you can see a separation here between females and males. Now you can see here there's a separation here based on age also, which is the x-axis, and the deviation of strong symptoms versus weak symptoms in women is at the age of 50, maybe indicating something related to hormones, and if you look here at the separation in males, it is at the age of about 30. This is ongoing studies, we're still analyzing the data. Yazid from the lab, together with a team at Soraski Medical Center with Dr. Ahuva Meilik and Dr. Amos Adler, analyzed bloodstream infections. We looked at EMRs here at 8,000 patients, all of them with a positive bloodstream infection, and a certain percent of them will die either during hospitalization or 30 days later. A simple machine learning was able to study for several years what features would indicate severe infection or death from this infection. This is a training set. A test case was carried out in 2019, and now we are applying it here. You can see the training set, the AUC here curves of the training set, the test sets, and now we're applying it there, and we're doing the validation set. If you want to see the different features here, I saw albumin on a few slides here this morning in several talks, and we also see it here in our slides as a separating feature. So to summarize my talk, we try to look at the clinical data. We speak with clinicians. We try to help the clinical team to reach significant insights or decisions based on bioinformatics we carried out and the biology we carry out in our lab. This is the chef teams. So they also cook in the lab and they cook outside the lab. These are the projects and the people working on the projects I talked about here. And these are our collaborators. Thank you.
Thank you, Noam. Stimulating talk.
Hi, thanks for the talk. So you showed plentiful applications of taking whole exome or whole genome sequencing and applying it to different diseases. How expensive or how feasible is it to put a sequencing machine in the clinic and how to sequence the patients?
Every large hospital and also the HMOs in Israel, all of them have an NGS, and most of them have the best NGS. They have the Novasec. It's cheap. It's cheap to do whole genome sequencing. It's about $600 and saves a lot of time and money of other tests.
Last quick question. Okay, so thanks again.
Thank you.